Okay, I think I'm ready. Are you guys ready? I'm gonna get the screen dropped down momentarily. I typically never teach in this room. I have one time, so I don't really know what's going on. But I know what's going on in micro, I hope. Okay, welcome to microbiology. This is Principles of Microbiology. We have shifted it up the littlest bit. It used to be back in the day, there were two separate courses, introductory micro and general micro. Both courses were very similar. The labs were super similar. And we decided to streamline some material and merge it all together. So I know, as I look out at the audience, there are many people that aren't microbiology majors. This is something that's a stepping stone for one of your majors. In the general micro, which was more for microbiology majors, it also was a stepping stone for some others like pharmacy. And in turn, really kind of just a handful of people that were really thinking, holy cow, microbiology, I want it. We're gonna put you all together. There's also some benefits because in some instances, Sheldon.
Yes.
Could we get the screen down?
Yeah, I'll go find the handle.
Oh, we need a handle. Okay.
Yeah,
I figured out how to turn the projectors on, but okay. So there are potentials that as you are thinking about whatever major, maybe you shift some gears and decide something else. And so by having this together, if you happen to decide maybe microbiology is really what you do want, then you don't have to bounce back and take a course that's pretty much redundant. So it's a win-win. We have taught this merged together last summer, last semester. It's my third round. I feel good. I feel comfortable. So there are lots of details when we think about microbiology, a huge amount of information that you may feel bombarded with or overwhelmed with, but I hope that that's not how you truly feel at the end of the day, overwhelmed. So we want to think about some principles of microbiology, some details about understanding all of, a plethora of different types of microbes. We'll go through some details about taxonomy, how we group them up. I'm not going to go into great detail with that stuff because there's a lot of other material that I find more fascinating. Still like understanding a little bit about who we're talking about. We'll go through some details about some of their ecology. There is huge diversity when it comes to microbes in the environment or in our bodies, all sorts of different potentials for some of these to maybe make a very nice living. We'll go through some details about handfuls of different microbes and how they go about their business, feeding on things like we might think about, or feeding on things completely different. There's all sorts of potentials with that, and we'll go through some metabolism. I assume you have all had some biology and thought a bit about stuff like metabolism, DNA replication, things like that. We're still going to touch on a bit as we move through with the focus on microbes. And ultimately, when it comes to some of these things, microbes really are kind of the key organisms that we think about in truly getting a firm understanding about the world around us. What I truly love is disease. if I could spend the entire semester just talking about disease, I probably would, but I won't. We will have a smattering of some disease as we move through here, but really the last chunk of material focuses on infectious disease. So you'll get a little bit of it. We'll also talk about another thing that I think is really pretty fascinating. We'll go through some details about immunology, thinking about our bodies and how we have potentials to deal with some of these organisms. But what I think is super cool is kind of this arms race between the microbes and ourselves, and how they, just as we evolve strategies to try to combat them, they evolve strategies to try to stick around and take hold and truly establish that disease process. Some organisms decide, I'm going to live right inside of this macrophage that should be doing the job of killing me. I'm going to live right in there. Brilliant. So we'll hit on some of that, and we'll talk some details about some of the applications, because it's not just all disease, even though that's kind of what I like to think. There's all sorts of things that we think about with microbiology, and potentially consuming stuff where we're relying upon some sort of microbial interactions to produce that stuff. I know some of you are yogurt eaters. Good stuff. All right. I put this on here, but this shouldn't matter because you're in here because you've already met the prerequisites, I assume. Some chemistry and biology, either 1101 or 2227. I know I've got a diversity of students in here, some thinking more health related and probably having some 2227. Others maybe going down some biology route, not necessarily micro yet. And maybe you took general biology. So either way, no stick.
Oh, yeah.
We might just have to shoot it right off the wall. That's all right.
We can do that.
It's fine. If you can't find it, it's all good. I assume. Is that all good? Good enough. You're looking at the other things. All right. So the textbook, hopefully you have looked a little bit at some of this material. Oh, yes.
This isn't the good stick, but
It looks like it could do some damage, though.
it sure could.
So the text is free from openstacks.org, I believe. Yes, there it is. You can click on the link. There are all sorts of different textbooks, many of them very expensive. OpenStax free, I like that. And the way that it's put together is very similar to textbooks that I like to teach with. Microbiology can be divided into either a taxonomic approach, first bit up in here, where we think about all sorts of different organisms, grouping them up, and then we'll talk a bit about disease, but it's thrown in there all throughout. Others do a systematic approach, which is what I prefer. Many times we see some bacteria repeated in different body sites, but you're focusing on the GI tract or on the skin, something like that. And I feel like, at least from my standpoint, that's kind of easier to wrap my head around all of the microbes from a disease standpoint. All right. So I like it. I like the way it's formatted. There is a lot of information in the book. The PowerPoints that I have that go with this are a little light, and I will try to ensure that there's ample material filled in there. And sometimes I will also flip over to OpenStax because there's some material in there. Tables, especially that typically didn't get put over into the PowerPoint. And I've tried a couple of times and I can't just copy them over there. I can copy images, but the tables doesn't play very well. And I probably need Sheldon. He could probably do it real quick. Okay, so you can access this through this link. Hopefully you have already looked at it, bookmarked it, so you can start going. Another thing that we have in here, and this one is not free. Packback is an online writing platform. I have used it in multiple classes for a couple of years. I think it's really good. There are boatloads of different things. I have used textbooks with online homework stuff. Some of that's good, some of it's not. I've done stuff where it's just straight up tests, nothing else. There's always issues. Some students would prefer just a test. Lots of students would like something else, so there's a little bit of a buffer. It truly kind of depends. To spend a boatload of time on homework assignments that maybe aren't super beneficial, really just kind of busy work, and making you spend $200 so you can do it, seems dumb to me. So I did that a little bit and decided when I saw this platform, maybe this is better. There are potentials for online forums. You could do stuff on Moodle. Very different than what Packback has going on. How many people have used Packback? A good chunk of you, probably 22, 27ers, because I know they use that in there. So a good chunk of you already sort of familiar with it. There are lots of opportunities with this platform to really kind of think a little bit outside of the box. As we start going through some specific concepts, maybe there's something that you're curious about, you come up with some different questions and post some stuff. So you will ask a question and you will respond to 2 other questions that have been asked. That's the way the Packback is set up. Every week, 5 o'clock deadline, not this week. The first Packback is due next Friday, to give everybody a little bit of time to get in and get stuff situated. If you are logging into this, as you move through, you should have received an email from Packback Friday, I think, inviting you to the community. If you didn't get it, this is the invitation right here, and the course lookup key, community lookup key, that gets you into my section. So once you're in there, it will bring you down to the bottom where you need to pay for it. I believe it's $30. It might be $35, somewhere right around there. You can sign up for it. You can pay a little bit later. If you have issues with paying, you can contact the help desk through Packback, wherever that's at, help at Packback, and they will set something up for you so that you can get in here and not miss any of the assignments. All right. So a huge amount of information in the text, huge amount of information on the syllabus. I like having more exams to cover the material rather than bombarding you with a dozen chapters and expecting you're going to be able to pull some information out of that. That seems overwhelming. So 6 exams. I've busted this book up into specific chunks or units. I'll show you the schedule in just one second. Each of these are worth 100 points apiece. Packback is worth 200 points, 800 total, but one of your exams I will drop so you can rest easy. If you miss an exam, that will be the one that's dropped. All right. The exams are going to be posted on Moodle. Take home, open book, open note, should be smooth sailing. All right. They will be opened, and I'll show you the schedule. Friday after lecture, they will open up, and they will be open until Monday at 5 p.m. to give you ample time to get in there, and a little bit of a window of time on Monday. If there's issues with anything, you still have time to deal with it. All right. So questions about that stuff? Feel good? Okay. There's that. I should have scrolled down. So typically multiple choice, little bit of matching and some true false. Short answer and fill in the blank, probably not so much. All right. The exams are really set up so I could roll in here and dish out a bunch of paper copies and you could take it in 50 minutes. That's how I feel. They're not, I don't think, more difficult being open book. I know there are some that are like, oh my God, it's open book. That's going to be brutal. I hope not. All right. You have access to your book. It should be very easy to move through there and get your information. I feel like that also is a benefit. All right. Many courses expect that you take stuff closed book and you just know all of this material. But deep down in the real world, people look stuff up. I know they do. All right. I've got kids taking them to the pediatrician. One of them sick. The doctor comes in after the nurse that really is doing all the work, right? After the doctor comes in, the doctor goes out and then they come back. What do they do when they go out? They look stuff up, right? That's what they're doing. There have been many times, our pediatrician knows that this is what I do, also knows that I geek out on disease. Many times that he would roll back into the room, textbook in hand, right? And I'm like, I knew it, right? We should have just stayed home. All right. There you go. All right. So I feel like that's the way it truly is. All right. Like this is a holdover from COVID. What's COVID? If you're sick, just stay home. I realized as I got in here, I didn't post a Zoom link on Moodle, but I will have one. The lectures are being recorded. I'm going to post all of the lectures onto Moodle. If you can't be here, they'll be there. I also have a handful of students that are sitting in OChem right now. All right. They will be watching the videos later. All right. I would appreciate it if you came to class, but if you can't, you have other opportunities, right? So if you're sick, stay home, all right? Wash your hands. That should be a no-brainer, and cover coughs and sneezes. All right, lastly, the italic stuff. This truly is where you find out you've just sold your soul. No one reads the fine print, right? All right. So Cozy, probably campus-wide, but definitely Cozy, has implemented an X grade. So if you just flat out don't show up and don't do anything, that's an X. That's like an F minus, all right? For financial aid, it's bad news, all right? So just do your assignments, take your tests. You're good to go, all right? I expect academic integrity when you are doing your exams. I would appreciate it if it's you that's doing your exam. That would be nice, right? I would hope that you don't work together. You've got your chance to use your book and use your notes. that should be all you truly need, all right? Not what did your friend get, right? All right. Don't make me go through academic dishonesty stuff. That's not fun. All right. How do I know? I know. All right. Accessibility. I have both campuses on here because typically I'm teaching distance delivery to Idaho Falls at the same time, not this semester, which is why we have been moved to a couple of different rooms. You may not even have recognized that any of that stuff happened, but we went from about 100 to about 140 students in the matter of about a week and a half. As soon as registration opened, we just had to keep bumping it up, keep adding labs. All right. With that said, no labs this week. You should have got a message about that as well. All right. Success, academic freedom, and land acknowledgement. This is a big one, at least in Pocatello. We are on the traditional lands of the Shoshone Paiute people, and we would like to pay respects for that. All right. No syllabus questions. I'm going to try to remember all of this stuff and share when I need to share. All right. Sharing is caring. Okay. So schedule real quick, just to break it down. The first 3 chapters is our first unit, a little bit about microbes. Truly, we could slam all 6 of them together because the diversity of these guys really kind of fits. But I think it's better to break it in half. All right. So that, then the next 3, then we'll go into some metabolism and genetics, the immunology, control of growth, and how our body deals with that. As we get into, immunology is the next bit. This is just the first bit of control, part of it. And then the last bit, which, like I said, I feel like I could really deal with this the whole semester. All of the different body sites, each of these is a respective chapter. And basically, when we get down to this point, it's going to be one body site per lecture as we move through there and give you an overview of disease. There is, if you're truly interested, pathogenic microbiology deals with this last chunk in the whole semester. So there's more if you truly want that. All right, and you can see the breakdown of all of the tests, the final exam. I'm going to open it up at the end of lecture, which would be right at the end of the last week of course. So you've got a little bigger window of time than the others. And I believe Wednesday is when that is set to close. I don't remember. All right. And that's based off of the final exam schedule because they want to control. All right. Questions about that? Feel good? All right. And all of this stuff is posted on Moodle. Okay. Oh, man. Video. I should switch that up. You're like, why am I right on there, right? I think the same as it's zooming in on me. I'm like, what the hell? All right. Now we're ready for business. Maybe. Okay. So, how many people in here are dental stuff, dental hygiene, dental? Good chunk of you. How about nursing? Another good chunk. Biology? Few. How about microbiology? Anybody thinking micro? One? Only one? Yet. All right. Okay. Anything else that I didn't hit? Business? All right. I figured probably not. All right. So lots of stuff. For the dental stuff, there's very little. I apologize. I'm going to talk about cavities for a second. All right. We'll go through a little bit. All right. Okay. We're ready. I'm ready. I almost took the whole time just to talk about syllabus. Okay. So the invisible world of microbes. Every time I see this image, I feel like this should be a commercial for Dawn dishwashing detergent, because deep down, this sucker's got a bottle of it down here. He's going to clean this turtle off with some dishwashing detergent, and it's got to be Dawn, right? I'm on YouTube right now. I should be getting some money, right? There it is. Free advertising. All right. So he's getting ready to clean this turtle. This was an oil spill in 2010 in the Gulf. And I think a disaster, but some pretty fascinating stuff when we think about it from a microbial standpoint. All right. There are all sorts of organisms that feed on, as I already kind of said this, feed on all sorts of different things. All right. Stuff that we might think of as flat out inhospitable. All right. We don't want that, right? I don't want to eat oil, but Alcanovorax does. All right. And after this spill, we saw a large bloom, if you will, of these bacteria thriving. And I think of it like, rolling into a restaurant and seeing a buffet as far as you can see. And you're just like, sweet, a taste of this, a taste of that, right? All of a sudden, these microbes that were struggling to survive have been taken to the buffet. Their fuel source is all over the place, and they skyrocketed, all right? And scientists ultimately trying to study these microbes and think about potentials for stuff like bioremediation, using some living organism to try to clean up our problems, basically, is really what we think about with that, all right? There's a lot of stuff that started going through my mind. I was like, oh man, that's brilliant. You could clean up all sorts of issues. And then I think about it a little bit deeper and wonder what would happen if these bacteria were able to truly be cut loose at that buffet. Do you think that they're going to just clean it up and be done with it? Or are they going to switch gears and do something different? Could they potentially go deeper and get right into these oil wells and start feeding at the source? I bet we don't want that, right? Lots of ecological implications when we think about things like bioremediation, all right? There are other things that I think about, not the same as bioremediation, but utilizing something to control something else, like maybe some insect to control some other insect that's a crop pest. Maybe that's a good idea. Maybe it's not. Lots of times we can think about microbes in those same instances and using them to do something that we would like. And then it makes you wonder what truly is the end result of that. So promise for sure, and some potential issues. So medical stuff. Here we've got a spinal tap, so a lumbar puncture, and collecting some spinal fluid. I believe, although this is a little grainy image, I believe this spinal fluid looks good, all right? Looks clear, all right? I think, all right? If it came out very dark or turbid, really cloudy, that would indicate something else, something going on, right? Potentially some sort of infection. And an infection in the nervous system, bad news, all right? We will touch on some details with that a bit later, all right? We will go through processes as we start thinking about some of these things, like how do we collect a sample? How do we culture it? How is it that we can get it to grow? And how do we ultimately differentiate what it is that we see? Sometimes, like maybe in the spinal tap, we might pull something out and it could very likely be only one type of organism, the infectious agent. But other body sites that we would think of as not necessarily sterile likely have a boatload of different players. If you think about maybe some GI issue, right? You got a lot of bacteria in your feces. A lot of the feces is bacteria in the first place, all right? How do you know who is the good guy and who is the bad guy? There's a lot of differentiation, a lot of specialized plates that we might employ in a microbiology lab to do some weeding, to get rid of some of those that we know should be there and focus in on the ones that likely shouldn't be there, right? So we'll go through lots of those details as we progress along. And a lot of that stuff we're going to think about before we really even get to the end infectious disease stuff, because it's super important, all right? When we think about the lab, there are lots of opportunities to get in there and start separating and isolating and growing bacteria. And we will show you handfuls of different types of plates and different types of media tubes that we can use for biochemical tests to help to differentiate specific organisms, all right? Okay. So shifting, I mentioned yogurt as we started. Bread is another one that is super important when we think about microbes in the production of it. Yeast like Saccharomyces cerevisiae is a yeast that is super common when we think about food production. Bread, frothy beverages, with a name like Cerevisiae makes me think cerveza, makes me think some beer, all right? Similar, but slightly different strains of these yeast. There are all sorts of different types of yeasts that we could employ for the production of different types of breads, to give subtle differences in frothy beverages, or even in the production of different types of wine. Different strains of yeasts that impart different characteristics and potentially alter the amounts of alcohol that could be produced if we're thinking beer or wine production, right? So the yeast are Saccharomyces. These are types of eukaryotic cells, single-celled microbes that have, like eukaryotes, membrane-bound nuclei and all sorts of organelles that our bacterial friends lack. Very different, all right? So this image, we could see some of the yeast cells, and technically you can see some details, nuclei, and maybe some other organelles within there that are showing up as little blebs on the surface of these cells. So when a yeast cell is added to the buffet, if you will, when we give it stuff that it would truly like, oftentimes the yeasts are freeze-dried, they're hanging out, but they're not growing. Once you add them to the dough, there's water, there's sugar, there's all of the sugars within the grains, within the wheat, that are available to these yeasts. And they start growing and replicating very rapidly, producing lots of gas bubbles, carbon dioxide in the process of its metabolism that makes the bread rise, right? If we're thinking frothy beverages, this is a similar sort of an idea, maybe different stuff going into the mix, but a similar idea. And those yeast cells start to grow. But once we get them growing really well, we cork it off and basically take it so that it went from a very rich oxygen environment to pretty much none. And those yeast cells have the opportunity to grow either in the presence or without oxygen. And when they have no oxygen, then they ferment and they start producing alcohol. They're still bubbling away, but instead of it just being carbon dioxide that's given off, alcohol is also a byproduct, all right? And we'll talk a little bit about some of that later on, all right? All right, and I do have a couple of links in here to a couple of YouTube videos, but I'm gonna skip over that right now, just so I can progress along the littlest bit. So when it comes to microbiology, a lot of our thinking really is disease, things that may be making us ill or mechanisms that we might be able to employ to help keep us healthy one way or another, all right? And we know this from all sorts of disease that plagued us for forever, right? How do we deal with it? One of the main ideas that we might think about, and something that really kind of seems like just sort of a no-brainer. We take a lot of this stuff for granted. You need a drink of water, you go to the tap, maybe in Pocatello you do. You go to the tap and you get yourself a glass of water and you don't think anything about it. All fine and good, all right? But back in the day, that wasn't necessarily the case, all right? And oftentimes drinking water could make you ill, all right? That's why there was so much beer. Just drink the beer. And ultimately, that's really not really a joke. Lots of times that was a good mechanism to make it so that water wasn't so bad as we fermented and produced a bit of alcohol within there, making it a little more consumable, if you will. All right. Not that I'm recommending you all just turn to beer. All right. Probably not the best advice, I would say. So here we could see Cloaca Maxima. This is the greatest Roman engineering, stolen from someone else, I'm sure. Greeks, I think so. So really pretty impressive, all right, with the aqueducts that they built, bringing fresh water into the city, and in turn, some water for the sewer system, draining it away. That water's not the same water that you're consuming. That wasn't always the case, all right? Take home message, don't drink where you poop, right? That's probably bad news. Quit pooping in the street, right? Bad news, all right? So this mechanism, getting fresh water and dealing with waste, dealing with the sewage, was huge and a big stepping stone for the advancement of civilization, truly, when we think about these details, setting the stage for people to start being a little bit healthier, all right? A little bit, all right? And there are all sorts of individuals before we start thinking about this that really kind of set the stage, thinking a little bit about disease, about microbes. Before we ever knew what microbes were in the first place, there must be something that's causing this, all right? There were plenty of others that thought of things as this kind of miasma approach, foul smelling air, things that arise spontaneously. I assume you took some biology, you probably talked a bit about spontaneous generation and going through the process of really disproving that. We're going to talk a little bit about that in here as well. Microbiologists, like our good friend Louis Pasteur, were critical in setting up experiments to truly show that microbes were the result of this foul-looking broth and not something that just arose spontaneously. And that was something that didn't happen overnight, all right? It took centuries to truly disprove the thought, the belief of spontaneous generation, right? You get stuck in your ways and it becomes very hard to change your mind. That's what we've always done. That's what I know. That's the way it is, right? Maybe it's not. One thing that I find impressive as we look at this latrine, I want a bit of privacy, right? I'd like some stalls going on, right? This, not like that, right? You'd just be talking to your friends as you do your business, right? Little bit different, all right? And maybe that's how you really are, right? You want a little communication as you're in there, right? I don't think you should, right? All right. So I said others. Hippocrates, the father of Western medicine, he believed diseases had natural causes, not supernatural causes. And he was huge in trying to solidify understandings of disease process before we ever were able to look through a microscope and see microbes in the first place, right? Thucydides observed, and I believe, if I recall properly, he survived as well, the Athenian plague, and recognized survivors were immune to the infection. They could deal with loved ones that were ill, potentially watching them die, and nothing happening to them. So first kind of hints of immunity and body's mechanisms to try to combat whatever it may be. And with this, I think this is maybe some Wikipedia. It is. There are all sorts of plagues that have plagued humanity. And many of these, from way back in the day, they've been able to kind of pinpoint and figure out this is most likely what it was. The Athenian plague, they don't know, right? They truly have no clue what the culprit was that spurred this. I think that's interesting. All right. Varro proposed, this is our first hint of some microbes. The disease could be caused by some minute creatures, all right? Microbes, but yet to be given that name, all right? So setting the stage to start thinking away from the supernatural and to the living world, right? Oh, Louis Pasteur. So he did more than pasteurize your milk, friends. Lots of stuff, all right? All sorts of innovations. He was credited for saving the silk industry in the French colonies with a pathogen that was plaguing the silkworm moths, or the larvae, the worms, one bit, all right? And pasteurization, spontaneous generation, all sorts of details, all right? Numerous advances in understanding disease processes as did Robert Koch. The two together, we really kind of think of as fathers of microbiology. He identified lots of different organisms. Anthrax was one of the first that he was working with, cholera and tuberculosis. And these infectious organisms, he was able to go through a process that technically is still utilized today. We call it Koch's postulates. We will touch a little bit on it later on down the road. But the idea with this is to cultivate the organism. So anthrax, there's a lot of different ways anthrax can infect, but a skin infection, pretty common. And these situations like handling animals, so dealing with livestock, set the stage for people to be more likely to become infected, right? They'd get a wound on their hand or on their arm, a scrape or a scratch, something like that. And the spores of the bacteria could get into that wound. They start to grow, they germinate, they start growing and replicating, and it causes disease. So you get a big ulcer, a big wound, and you could scrape some of that and put it onto something to allow it to grow. Back then, potato slices were very common. And you'd slice that, swab your stuff on it, and allow it to grow. Then we start thinking about agar and petri dishes and a little bit different, and Koch fits right into that as well. But initially, trying to grow it up, all right? You grow it, you take that from your slice of potato, and maybe you get a lab animal, a little mouse, and you make a wound, a little scratch on it. You inoculate it with what you grew on that potato slice. You let the mouse incubate it up, if you will, and it develops the same lesion. You swab it off of it, put it onto another potato, same potato looking slices, same infection looking slices. This thing off of this guy caused this thing on this mouse, done deal. This is the infectious agent. That's Koch's postulates, all right? Lots of different infectious diseases go through that process to truly solidify this is the culprit, all right? There are some that we haven't done Koch's postulates with. And I'll leave you with that. That's the perfect stopping point. Farewell, good day. See you all Wednesday.